This video demonstrates the procedure for photoconversion of the Styryl dye FM143 to study synaptic vesicle pools in Drosophila melanogaster. First, Drosophila larvae are dissected to expose the ventral muscles, which constitute the neuromuscular junction, or NMJ, preparation. The preparations are incubated in buffer containing FM dye, shown here in gray, labeling cellular membrane. They are then electrically or chemically stimulated to induce vesicle fusion. Vesicle membrane internalized following stimulated exocytosis will be stained with FM dye. The extracellular FM dye is then washed, removing it from the plasma membrane, and the tissue is fixed. Under continuous high-intensity illumination, the dye is completely bleached, and a black color appears due to diaminobenzidine precipitation. This precipitate can then be visualized by transmission EM.
Hi, I'm Filippo Passo from the laboratory of Silvio Rizzoli at the European Neuroscience Institute in Göttingen, Germany. Today, we will show you a procedure for FM photoconversion in Drosophila neuromuscular junctions. We use this procedure in our laboratory to study synaptic vesicle recycling. Okay, so let's get started.
Working under a dissecting microscope, begin by pinning a Drosophila larva, dorsal side up, in a Silgard-coated dish. Cover it in Drosophila saline. Using a syringe needle and fine dissection scissors, section the dorsal side longitudinally. Then remove the internal organs. Next, stretch the preparation and again pin it to the Silgard dish. Several ventral muscles can then be used. To protect the FM dyes used in this procedure from bleaching, this procedure should be performed under low light conditions. Cover the pinned Drosophila preparation with Drosophila saline containing 90 millimolar potassium chloride and 10 micromolar FM143 dye to chemically stimulate the nerves. Incubate for 1 minute at room temperature. Following stimulation, remove the stimulating solution and submerge the dish with the pinned Drosophila preparation in a beaker filled with 100 milliliters of Drosophila saline 2 times to remove the extracellular FM143 dye. Working under a chemical safety hood, fix the neuromuscular junction preparation in 2.5% glutaraldehyde in phosphate buffered saline, or PBS, for 45 minutes at room temperature. Wash the preparation once with PBS. Then, remove it from the Silgard dish and submerge it in 100 millimolar ammonium chloride for 15 minutes to quench the free aldehyde groups of the remaining glutaraldehyde fixative. Using normal PBS, wash the ammonium chloride solution. Place the neuromuscular junction preparation in a new Silgard dish and submerge it in filtered 1.5 mg per milliliter diaminobenzidine, or DAB, in PBS and incubate for 30 minutes at 4 degrees Celsius. Position the sample under a fluorescent microscope equipped with a mercury lamp light source with a lamp housing containing a back mirror. Using a relatively low magnification dry objective, find the sample. Then, using a normal blue excitation green fluorescence filter cube, bring the fluorescent signal into focus. The neuromuscular junctions appear as bright spots on the muscles. Next, using the same fluorescence filter set, illuminate the sample at maximum intensity until the FM dye is completely bleached. Using a 20x objective lens, 30 to 45 minutes should be sufficient. However, the time for photoconversion varies depending on the intensity of illumination strength, the penetration of DAB into the preparation, and the objective lens used. Following illumination, examine the sample under transmission light. If photoconversion has taken place, a dark brown precipitate should be seen. Return to the dissection scope, then use scissors to cut the photoconverted spot from the larva. Return to the dissection scope, then use scissors to cut the photoconverted spot from the larva. Now place the sample in a microfuge tube containing PBS. Working under a chemical safety hood and wearing appropriate safety equipment, including gloves and eye protection, prepare 300 microliters of 1% osmium tetroxide per sample. Now, place the sample in a plastic-topped glass flask and add the prepared osmium tetroxide solution to post-fix the preparation. While the sample is incubating, prepare separate solutions containing 30, 50, 70, 90, 95, and 100% ethanol. Wash the sample 4 to 5 times with PBS for 5 minutes each. After each wash, use a plastic Pasteur pipette to transfer the sample to a new flask. After the last wash, transfer the sample to a clean glass flask. Next, dehydrate the sample by placing it in the series of ethanol solutions. Begin by incubating in 1 milliliter of 30% ethanol. Then, after 5 minutes, remove the ethanol using a Pasteur pipette and replace it with 1 milliliter of 50% ethanol. Continue with the dehydration, incubating the neuromuscular junction preparation in the 70, 90, and then 95% solutions. The sample will then be incubated in the 95% ethanol solution 2 more times, then incubated in 100% ethanol 3 times. Following dehydration, add 2 milliliters of 1:1 epin resin to ethanol and place the sample on a rotator. Incubate 2 to 4 hours at room temperature. Following rotation, add 100% epin and place the sample in an open flask to allow the remaining ethanol to evaporate for 4 to 6 hours. Pour fresh epin into a mold. Add the preparations and incubate at 60 degrees Celsius for 36 hours. When the epin-embedded preparations are ready, use an ultramicrotome to prepare 50 to 80 nanometer thin sections. Using a diamond knife, the samples are cut in thin sections. The sections float on the water bath of the knife and can be afterwards picked up on normal EM grids. The preparations are now ready to image using conventional EM procedures. Isolated neuromuscular junctions from Drosophila melanogaster were stimulated using high potassium, and internalized membranes were stained using FM143. As shown here, the fluorescence of a nerve terminal can be observed using a conventional epifluorescence microscope. Under continuous illumination, the dye is completely bleached, and when illumination continues, a black color appears due to diaminobenzidine precipitation. This nerve terminal contains synaptic vesicles, but none of them are photoconverted. This might be caused by not having enough illumination or poor diaminobenzidine penetration in the tissue. By contrast, too much illumination results in an overall dark terminal where no vesicles or organelles are distinguishable. With the appropriate amount of illumination, photoconversion can be seen. This micrograph shows a synaptic bouton in which a pool of dark vesicles are seen, indicating FM photoconversion.
We have just shown you how to photoconvert synaptic vesicles labeled with FM dye. When doing this procedure, it's important to remember to always carefully calibrate the illumination times for the conversion step. So that's it. Thanks for watching. Good luck with your experiments.